Welcome back to this channel, and this is the second video on electron transport system. And today basically we'll be discussing about the ATP synthesis. So in the previous video, you can find the link over here in the upper right corner of your screen. So we have discussed about this electron transport chain. We have discussed about complex 1, 2, 3, 4, and today we'll be discussing about this fifth complex, that is complex 5 or ATP synthesis. Now, as you can see over here, this is the case of eukaryotes. I have already mentioned in my previous video that electron transport chain is present in the inner mitochondrial membrane or in the inner mitochondrial membrane in case of eukaryotes and plasma membrane in case of prokaryotes. So this part we have already discussed in case of eukaryotes, and today, for the simplicity, we'll discuss the ATP synthesis about in case of prokaryotes. Okay, so this is, you can see, this is the prokaryotic plasma membrane. This is outside of the cell. This is the cytoplasm. This is the ATP synthesis, and this is the this is the integral membrane part, and this is the part which is present in the cytoplasm. Now, this is in case of prokaryotes, but in case of eukaryotes, all these things are same. Just you have to remember the outside is actually the intermembrane space in case of eukaryotes, and the cytoplasm is actually the mitochondrial matrix in case of eukaryotes. So you can see over here, this is the outside. This part is the intermembrane space, and this is the integral membrane part, and this is the globular part of this ATP synthesis, and ATP is being synthesized in the mitochondrial matrix. And in case of prokaryotes, ATP is being synthesized in the cytoplasm. So this is the simple difference. Okay. Hope that will not create a major problem in your understanding. So let's deep dive. So as you can see that complex 5, that means, yes, obviously ATP synthesis is known as complex 5 in case of electron transport chain, and we have two parts for ATP synthesis. One is the F0 part, another one is the F1 part. So that's why this is also known as F0 F1 ATP synthesis, because there is an F0 part and an F1 part. Now in the F0 part, you can see there are 12 to 14 C subunit, 4 to 6 A subunit, and there is also a B subunit which is kind of linked in here. But since we this particular picture is kind of a slicing of this F1 part, so it has been sliced out, but this is actually linked, and so this B part is kind of joining this F1 and F0 part along with this gamma stock. Okay, so this is one part. This is the F0 part, which contains A, B, and C subunits. There are 10 to 12 C subunits, 4 to 6 A subunit, and there is one B subunit. And in the F1 part, there are 3 alpha subunits, 3 beta subunit, and 1 gamma subunit. 3 alpha and 3 beta subunits are present in alternative structures, in alternative form. So this is alpha, this is beta, this is alpha, this is beta, this is alpha, and this is beta. And this gamma is kind of a central stock which can connect this F1 and F0. So there are two major parts which connects F1 and F0. One is gamma, another one is beta or B. Okay, and this whole thing is F1 F0 ATPase or ATP synthesis. Rather, now what happens is the proton concentration is present in this part. So this is the part where high concentration of proton is present, and this is the cytoplasm of prokaryotes where there is less amount or low concentration of proton. So this creates a proton gradient. Okay, and this proton gradient is also, we can say, there is an electrochemical proton gradient. Why electrochemical? Because as there is proton gradation, there is also a gradation of positive charge. H plus ion more in this case means in this part means positive ions are more in this part, and negative ions are more in this part. Or we can say there is high positivity outside and low positivity inside. Okay, so that's why there is an electrical gradient. And why chemical gradient? Because obviously H plus and high means pH is less, so acidic outside and basic inside. H plus is less in this cytoplasm means pH is high, that means basic pH, and H plus is high in outside means pH is low, means acidic outside. So acidic outside and basic inside. So both of this, acidic outside, basic inside, and more positivity in outside and less positive inside, both of this, they form the electrochemical proton gradient. And this electrochemical proton gradient is very necessary or very needed for converting this ADP and PI into ATP. Okay, so this is the overall structure. Hope you find it easy. So this is ATP synthesis, and next we'll discuss about how ATP synthesis work. So as I have already mentioned that in the F1, see, in this F1 part, there are 3 alpha subunits and 3 beta subunits with 1 gamma stock. These alpha subunits have their structural role, but these beta subunits plays the most important role because these beta subunits are the major functional part. Okay, so what happens is, yes, this is the nice picture. So what happens in this case is, see, we have these beta subunits in here. Okay, so there's 1, 2, and 3 beta subunits, and these beta subunits has their own configuration. So there is an open configuration, there is a tight configuration, and there is a loose configuration. Why these configurations are different? Because they have different binding affinity for their own substrates. Okay, this open conformation is where ADP and PI can come, and the produced ATP can be removed from this open structure. So this is like open for substrate and open for product release also. Loose configuration is when ADP and PI needs to get reacted so that the ATP can be produced. And this tight configuration is needed to just hold the ATP for some time, for some stability, or for a specific period of time until the whole reaction is completed. Until the ATP get really stabilized. So that's what the function of the tight configuration is. So whenever there is an open configuration, that means ATP and PI can bind and ATP can get released. This tight configuration means whenever there is a tight configuration, there is the ATP cannot be released, but the ATP can get stabilized. That means ADP and PI in this tight configuration, ADP and PI can react and ATP can get stabilized. And in the loose configuration, ADP and PI can react but loosely. Now what happens is this is very important. This is the gamma, this is the stalk, and it can rotate. So this rotation of gamma will also change the structural configuration of this beta subunit. So let's say there is some energy, and that energy is being provided by what? By this electrochemical proton gradient. This is the energy source over here. This is the energy source. Okay, from this electrochemical proton gradient, this energy is coming, and rotation of this gamma subunit cause conformational changes at the catalytic sites, which release the ATP. Let's see how. Now this loose configuration, there is a rotation. Okay, in this part there is a rotation like this. There is a rotation. So due to this rotation, this loose configuration comes over here. Now you can see this is the loose configuration. This open configuration comes in here, and this is the open configuration, and tight configuration goes like this. So here now we have tight configuration. Now as I have mentioned, the open configuration is open for substrate acceptability, also for release of the product. That means now ATP will get released from this open configuration. These ADP and PI, which were bound at the loose configuration, which were reacting loosely, are now reacting in tight configuration. That means ADP and PI are reacting to form the ATP, and that ATP will remain stabilized until again that ATP is facing the open configuration. So that is the function of ATP, tight configuration. Hope that makes sense. And again, we have loose configuration and ADP and PI. This ADP and PI, which was just introduced in the previous step, is now facing the loose configuration. That means ADP and PI have started interacting to each other loosely. And then there is a second thing, or second reaction occurred. Second step occurred. That is, now this open configuration is now empty because ATP has already been released, and so new ADP and PI will come and with this open form or open configuration. And in this tight configuration where ADP and PI were reacting tightly, now ATP has been produced and it is now stabilizing and is now ready for being released. In the next phase, whenever there is another rotation and open configuration comes from here to here, this ATP will be released. Till now, in this stage, this ATP has been synthesized and is remain bound tightly to this configuration. And again, in this part, ATP and PI have started interacting with each other loosely. So this is the way. This is how this ATP synthesis works. This is how the gamma subunit rotate, and there is a structural conformation change in the beta subunits. And then each time open configuration face the ATP is released, and new ATP and PI get inserted or get attached to this open conformation. So in each case, in each step, ADP and PI first is introduced into this open configuration, then it will undergo a loose interaction, then finally it will undergo a tight interaction, then it will again face the open configuration. So this is a cyclic structure, and this cyclic way this ATP synthesis goes on. Now here's a very interesting point, that the F1 ATP synthase can perform its ligase function. That means this ADP plus PI interaction function, like interacting ADP with PI, without proton flow into matrix. However, the release of ATP requires flow of proton through the membrane. So the flow of proton that we have discussed in our previous video, that there is a flow of proton, and that flow of proton is very much needed for ATP synthesis. We now know that for this ligation function to interact ADP and PI, this ATP synthesis doesn't require that proton flow. That proton flow is actually required so that the ATP can get released. So that is a very important and interesting point. Okay, please remember this point. Now we'll discuss something more about this ATP synthesis. So this is a very, very common structure, very simple structure. So this is ATP synthesis. H plus is in the outside. This is the F0, this is the F1. H plus goes in, and ADP and PI interact, and it forms ATP. Okay. Now for ATP synthesis, two components are very much needed. One is pH gradient, another one is ionic gradient. Obviously, the H plus should be more outside this membrane, and there should be ionic gradient. That means positive ion, the number of positive ions should be more at the outside of this membrane. Okay. So between the intermembrane space and mitochondrial matrix, again, if we just think about the eukaryotes, this is the inner mitochondrial membrane, this is the inner mitochondrial membrane space, this is the mitochondrial matrix, and the difference should be this pH gradient and ionic gradient should be between what? Between intermembrane space and mitochondrial matrix. This is for eukaryotes, and between the outside of the cell and cytoplasm in case of prokaryotes. Okay, as we have seen over here, outside of the cell and cytoplasm, this pH gradient or the ionic gradient should be there. Okay, so positive out and negative in, I've already discussed. Positive outside, H plus is more outside, so positive outside, negative inside. And acidic out and basic in, H plus is more, that means pH is less, so acidic is outside and basic is inside. So these things are needed for all these things. They provide sufficient amount of energy for ATP synthesis. So that is the major concept of ATP synthesis and ATP synthesis. If you find this content helpful, please make sure to like, share, comment, and subscribe. Follow for more. See you in the next video. Bye.